Good morning, good afternoon, and welcome everyone to today's live broadcast, Biomedical Applications of Raman Spectroscopy. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Metrohm USA. For more information about our sponsor, please visit their website at metrohm.com. So let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, please click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd like to now introduce our presenters, Kristen Frano, Applications Manager at B&W Tek, and Dr. Adam Hopkins, Spectroscopy Product Manager for Metrohm USA. For a complete biography on our presenters, please visit the Biography tab at the top of your screen. Kristen and Adam, you may now begin your presentations. Welcome.
Thanks, Alexis, and thanks to those who are joining in today. So we're really excited to be talking about this topic, which like Alexis said, is biomedical applications of Raman spectroscopy. So first I'll give a brief introduction to some of the basic principles of Raman spectroscopy, and then we'll dive a little bit deeper into discussing some examples of biomedical applications that our systems have been used for recently. And then Adam will give us an overview of the Metrohm Raman spectroscopy portfolio. So when we talk about spectroscopy, all we're really talking about here is the interaction of some kind of light with some kind of matter. And in that interaction, we learn some information about our sample of matter. And we get that in the form of an output, which in spectroscopy is a graph that we call a spectrum. So you can see that little picture of the Raman spectrum there. So there's the output that we get, and then there's the actual information that we can interpret from the output that can help us solve the problems that we're trying to answer. So from a Raman spectrum, we can get structural information about our sample that we can use to do either qualitative or quantitative analysis. So qualitative analysis is based off of the band positions of the spectrum. So Raman spectroscopy is a type of vibrational spectroscopy in which we're probing the vibration groups that are present in our sample. So we're looking at the actual chemical bonds in our sample. So because each functional group has its own kind of characteristic frequencies at which it vibrates, the Raman spectrum can act as a molecular fingerprint that we can use to identify a sample. So in this example, you can see the spectrum of the 1-butanol and the 4-isobutyl acetylphenone. That's the red spectrum. And you can see that because they're different molecules, they have distinct Raman spectra that we can use to help identify them. And this is a big advantage because this allows us to use spectral libraries to identify either unknown samples or to verify the identity of known materials. And also, shifts in the peak positions in our Raman spectrum can give us some information about changes that are happening in our sample matrix. For a quantitative analysis, the intensity of our measured peaks that we see is proportional to the amount of the analyte that's present in our sample matrix. So we can use the peak heights that we see to directly correlate the concentration of the analyte that we have in our sample. So we can do quantitative analysis based on peak intensities. just a little bit of some historical background information. So Raman spectroscopy is based off of the phenomenon of Raman scattering, which was first observed by C.V. Raman, who's pictured here, in 1928. So in his experiment, he isolated a violet light and was able to pass it through a liquid sample. So that liquid sample scattered the radiation, and he observed two types of light that was scattered, one that was also violet light, and then one that was scattered green light. And so if you look at our little electromagnetic spectrum at the bottom left, we can see that the green light is at a lower energy than the violet light. And so we have two types of scattering here, one that's at the same energy as our incident light, and one that's at a different energy. And it's this different energy scattering that we're interested here with Raman spectroscopy. So this is just a drawing of the Raman effect. So we have our incident energy that comes in and excites the bonds from our molecule. And we see our two types of energy that's scattered here. So we see our elastic scattering, which is our unchanged scattering, which is also called Rayleigh scattering. So this is at the same energy as the energy of our excitation source. And then we also have our Raman scattering that occurs. So that's our inelastic scattering, which is scattering that's happening at a different energy than the energy of our excitation source. So it's important to note here that Raman is an inherently insensitive analytical technique. So for a lot of molecules, it's difficult for us to reach really low limits of detection with Raman. And so it's for this reason that we need a really strong and stable light source that can build up the amount of photons that are going into our samples so that we can build up a strong signal of Raman scattered photons. So typically about 1 out of every million incident photons are Raman scattered. So a Raman spectrum is plotted with intensity on our y-axis and Raman shift on our x-axis. So the Raman shift is calculated by taking the difference between the energy of our incident light and the energy of our scattered photons. And so we measure the Raman shift in inverse centimeters or wave numbers. And the bands that are plotted represent the vibrational modes of our molecule. So these correspond to the different functional groups that make up the molecule. So in this example, we have a spectrum of cyclohexane, which is our purple spectrum, and a spectrum of collected from benzene, which is kind of that bright green spectrum here. And looking at the two structures of those molecules, they may seem somewhat similar, but due to the differences in their chemical bonding, right, we have our aromatic ring for benzene, so we have those carbon-carbon double bonds. So based on the differences of their actual chemical bonding, we see very distinct Raman spectra that we can use to do some sort of qualitative measurements off of. So just a couple of things that are important to note about Raman spectra. So they are independent of the laser excitation wavelength that is used. And so for instance, to the right here, we have a spectrum, or a set of spectra collected from toluene. And so these spectra are all collected with 3 different laser excitation wavelengths. So 532, 785, and 1064 nanometer lasers. But as you can see, the peak positions remain the same throughout the set of spectra, regardless of what laser is used. And so a question that we get asked really often is, well, okay, what laser should I use for my experiment? That's really going to depend on the type of sample that you're looking at, as well as some of the kind of analytical needs of the experiment as well. In regards to Raman intensity, so the Raman scattering strength is proportional to the energy of the laser that's used. So this means that the higher energy laser we use, the more intense our Raman spectrum will be, which also means that the longer wavelength that we use, the lower intensity our spectrum will be because it's a lower energy laser. So because of that, you might think that we should always use really high energy lasers so we can get really good Raman signal all the time. But one caveat here is that if we do use a higher energy laser, that it's possible that that could cause some fluorescence issues for us in our Raman spectra. So fluorescence is a competing technique. It's an emission technique, so the molecule is excited and then emits photons that are fluorescence. And fluorescence is a much more efficient technique than Raman is. And so if we excite the fluorescence in our molecule, it's possible that that could overwhelm the Raman signal that we're trying to observe. And then in that case, we won't get any peaks that we're able to do either qualitative or quantitative analysis with. So for instance, the example that I have is Raman spectra collected from sesame seed oil using 3 different laser excitations. Sesame seed oil is a really very dark brown oil, and it's because of that that we see some of the spectra, specifically the ones collected with 532 nanometer laser and a 785 nanometer laser. In those cases, the spectra are basically completely overwhelmed by the fluorescence that's generated. So we don't really see any of the Raman scattering that we're interested in. And it's not until we shift to using a longer wavelength, like a 1064 nanometer wavelength, that we're kind of able to avoid that fluorescence region, and then we're able to observe the Raman scattering from the sample that then we can do analysis with. So over the past few decades, there's really been a big push for Raman to enter the biomedical field. And the reason for that is that there's a lot of biomolecules that are Raman active. So these have functional groups that have, they have vibrations in their Raman spectra, so we can use them to do different types of studies. So tissue components like lipids and proteins and fatty acids and nucleic acids all have Raman signal that we can use to study them. And so the important regions for these materials come down to kind of 4 main regions. So one is our hydrogen stretching region from about 2,500 to 4,000 wave numbers. This may be important to study, carbon-hydrogen stretching in proteins. Triple bonds are found from 2,000 to 2,500. There's not a lot of really important triple bonds in biomedical analysis, but that's where they would be present. Double bonds, however, are very important in things like lipids, and those would be found from 1,500 to 2,000. And then from 600 to 1,500, we have kind of our fingerprint region where we really get the complex patterns that we can use as kind of an identifier for our specific molecule that we're looking at. And so Raman has a lot of advantages for biomedical analysis. So for example, Raman is a non-destructive technique, and it's very highly selective compared to even other spectroscopic techniques like near-infrared spectroscopy, where the bands that you get tend to be a lot more broader. Water is also a very weak Raman scatterer, so that's actually really good because we get very little interference when we use water as our solvent. So we can measure aqueous solutions, which is really a big advantage in the biomedical application. Also, the portability and the fiber optics of today's Raman systems really allow for flexible measurements in kind of a lot of non-laboratory settings. So you can actually see to the right here some of the examples of the fiber optic setups that our portable Raman has. So I think this is going to be really important kind of going forward because the trend in biomedicine is going more and more towards portable analysis and really looking for simple solutions that can be applied at what's called the point of need. So this might be either a clinical setting or an operating room, but this idea of portability is going to be really important moving forward. And although Raman is an inherently insensitive technique, we can actually use a variation of Raman spectroscopy called surface enhanced Raman spectroscopy, or SIRS, and this can increase the sensitivity of our signal by a factor of about 10 to the third to 10 to the seventh, depending on kind of what our experiment looks like. And this allows us to get down to trace levels of detection of biomaterials. And so we'll talk a little bit about SIRS in one of our examples. And I do just want to give a few examples of areas where our systems have been used for biomedical analysis recently. So the first example that I'll talk about is the classification of cancer tissue. So cancer is the leading cause of death worldwide. So you can understand why it's been such a growing application for Raman spectroscopy in recent years. So because healthy tissue and cancer tissue have kind of their own different biochemical compositions, we can actually use the differences in their Raman spectra to distinguish healthy tissue from cancer tissue. And so there's really a kind of a big push for portable Raman to be used in this space. Because like I was saying, then that means that your measurements don't have to be done in a lab. They can be made, for example, in an operating room. And so having the portable Raman system can kind of provide a real-time guidance for a surgeon. So this doesn't necessarily negate the need for pathology results, but it does help mitigate the reliance on those results because they may take 1 to 2 weeks to come back. And meanwhile, the patient is still kind of in limbo, not necessarily knowing the concrete results of their surgery. So in breast cancer lumpectomies, for example, the surgeon removes the cancerous tissue, and then what's called a margin analysis is performed on the sample. So I'll share some research from Harvey Mudd College in collaboration with the City of Hope Medical Center and the Kinohi Institute, all in California, where they used portable Raman to sort of classify the cancer margins in lumpectomy samples. So here's kind of a drawing which shows the definitions of positive and negative cancer margins. So if there is a cancer cells that are present at the edge of the tissue sample, that would indicate a positive margin test. So this indicates that it's likely that there's still cancer tissue that's left at the site of the lumpectomy. On the other hand, for a negative test, that means that there's no cancer tissue at the edge of the sample, so it's a likely indication that the surgeon was able to remove it all. And so in their experiment, they measured actually with 2 types of portable Raman systems, one with a 785 nanometer laser and one with a 1064 nanometer laser, because they were worried about fluorescence with the 785 nanometer system. They also were able to sample using kind of a variety of setups as you can see in the pictures that are shown, using the portable or using the fiber optic setup to measure in kind of a variety of ways. The top one using a probe support stand, also using a microscope, and then also using the handheld fiber optic probe as well. They were able to acquire 50 tissue samples, and while the pathologist was classifying the samples as either tissue samples, or normal samples, or cancerous samples, their Raman spectra were collected at the same time. So here we have data sets collected from two different laser excitation wavelengths. So the top is the 1064 nanometer data, and the bottom is the 785 nanometer data. And so it's very difficult to detect the differences here if you don't know what you're looking for. So especially in the 1064 nanometer data, because we have a little bit of fluorescence in the 785, but for the 1064 nanometer data, the healthy tissue spectra looks quite similar to the tumor tissue spectra. And so that's why we have to use chemometric analysis here to help us kind of parse out those very slight differences between the healthy tissue and the cancer tissue. And then at the left here, there is some peak designations for the functional groups that make up the components of the healthy tissue and the cancer tissue. So typically in cancer tissue, there's an increase in the contribution of the signal that's associated with the nucleic acids. So the DNA and the RNA signals are really prominent in the tumor tissue spectra. And also there's usually a decrease in the signal from the fatty acids compared with the healthy tissue. Then they were able to build a classification model out of the data using principal component analysis, or PCA, and linear discriminant analysis, or LDA. So the PCA is used for data reduction, and the LDA is used for the classification. So these are actually two really popular data treatments that are used really commonly in spectroscopy to do this type of classification analysis, especially on cancer tissue samples. So this is the, on the left is the 785 nanometer PCA data, and on the right is the LDA classification results. So for the classification, we can see that 10 out of the 10 tissue samples that the pathologist deemed as healthy tissue samples were correctly classified as healthy, and that 38 out of the 40 tumor tissue samples that they classified as tumorous were also predicted by the model to be tumor samples. So out of this classification model, they were able to get really accurate and sensitive results. So they proved that they can distinguish the cancer tissue from normal tissue. So they were also interested in assessing the margin analysis in some of the biopsy samples. So in this case, they're interested in kind of observing how the signal changes on a transition from healthy tissue to cancer tissue that can be found at the margins. So in the A microscopy image, the one to the left, we can see that there's different axes that are labeled, and so the one that's labeled 6 all the way down the center to 15 corresponds with the set of Raman spectra on the left. So that also starts from spot 6 all the way down to spot 15. So they measured the Raman spectra along this axis, and you can clearly see that as we kind of measure down that axis, that there's clearly a change in the Raman spectra that we see here, and this change represents the transition from healthy tissue to cancer tissue. And so in this case, if there's cancer tissue that's found at the edge of the sample, like as would be in spot 15 there, that would indicate a positive margin test. So I also wanted just to make clear that this type of analysis is often done with other types of cancer, so not just breast cancer, but often the method of data collection and data treatment using multivariate analysis is very similar. And I also wanted to show an example of kind of what a setup for an operating room might look like. So this is from a team at Ruhr University in Bochum, Germany. And here they have a portable Raman system that's set up on a cart that can be rolled from room to room for analysis. They also have a little custom sample holder for the tissue samples as well. So with the portable Raman system, you can place it on a cart and use the portability of that to bring it from room to room for different surgeries. So another area that I wanted to talk about a little bit is the use of Raman in vaccine manufacturing. So this is obviously a hot topic lately, especially this week. So we all know that vaccine manufacturing is a very long process. So beginning with raw material identification to the end with final lot release, it can take up to 22 months for an approved vaccine to be manufactured. And each kind of step along this process needs to comply with good manufacturing practices, which means that we need some analytical tools that can help identify and verify the different components of a vaccine along those various stages of the process. So these can include raw materials, intermediates, as well as the final vaccine formulation. So for raw materials for vaccines, these can include the active components, but as well as preservatives, adjuvants, which are used to boost the immune response of the vaccine, as well as stabilizers and antibiotics. But all of these materials are Raman active, and we can use their kind of Raman fingerprints to verify them as raw materials for vaccine manufacturing. So the spectra that I've shown on the right here are actually components of an IC31 adjuvant. And this is made up of a KLK peptide and an oligonucleotide, ODN1A. And so portable and handheld Raman here is really suited to measure these types of materials throughout the manufacturing process, but especially in the raw material identification stage. Another important material for vaccine manufacturing is cell culture media, because it's used to grow the microorganisms that go into the vaccine. So it's used to grow them under really controlled conditions, and the media provides the nutrients that they need to grow. So typically cell culture media is very complex. So they're usually composed of a mixture of amino acids, vitamins, glucose, and serums as well. So for raw material identification of cell culture media, handheld Raman is a really useful tool for this. So I wanted to show some data that my colleagues in Shanghai actually measured with a handheld system. So you can see they measured a few serum samples and also media samples. And they all have very good Raman spectra that we can use to do raw material verification from them. And also here I would say that a 1064 nanometer laser may be important to reduce that fluorescence that we may see from these types of samples. So the dextrose agar medium at the bottom and the bovine serum albumin at the bottom both fluoresce with 785 nanometer excitation. So 1064 is really the best solution for the cell culture media. So Raman can also be used in vaccine characterization. And so this is work that Marina Kirkatadze's group at Sanofi Pasteur in Toronto has done with their portable Raman system. So they use Raman to characterize a new tuberculosis vaccine candidate. So tuberculosis is one of the top 10 causes of death worldwide. And while there's a vaccine that exists for it, its efficacy is variable, especially in adults. So it's worth it to explore new options for a tuberculosis vaccine. So for this vaccine candidate, it's made by adsorbing an antigen protein, H4, to the IC31 adjuvant. But during this process, though, they think that there may be some structural changes to the H4 protein that occur upon that adsorption. And these structural changes are important for them to understand. So they want to study the structure of the protein while it's adsorbed to the adjuvant, because obviously if they adsorb it and then desorb it, that structure could then change again. So the H4 protein is made up of the antigens AG85B and TB10.4. So you can see their structures on the right, and they're primarily made up of alpha helices and beta sheets. And so they used Raman to measure the protein while it's adsorbed to the adjuvant. So that's the blue spectrum on the left-hand side. And they also measured the adjuvant by itself, which is the red spectrum, and they measured just the free H4 protein by itself, and that's the green spectrum. So in the spectrum of the free H4, there's a very weak signal that's consistent with beta sheet present at 1670 wave numbers. But when that H4 protein gets adsorbed to the adjuvant, which is the blue spectrum, we can see that that 1670 peak significantly increases compared to the spectrum, the green spectrum of the free H4 protein. So some of that signal can partially be attributed to the adjuvant, so the red spectrum, but not all of it. Some of it can be attributed to the conformational changes just of the protein itself upon adsorption. So their results indicate that upon adsorption to the adjuvant, the protein undergoes structural changes that involve kind of an increase in the formation of these beta sheets structurally. They did use some other analytical techniques that confirmed these findings, but I think this is a really good example of using qualitative changes in Raman data set to kind of help us understand the processes that are happening on a very fundamental level. And then I also wanted to touch briefly on how Raman has been used for disease detection, specifically with surface enhanced Raman scattering. So with disease detection, the trend here is to have really fast simple and accurate measurements at the point of need. So this could be in both first and third world countries. And so this is where portable and handheld Raman will really be an advantage here. And so for this example, we're using tuberculosis again, which is a disease for which diagnostic tests are sometimes inefficient insufficient due to the processing times that's associated with the tests, the costs, and the lack of portability of these tests. So again, here it's really worth it to explore new testing options. So disease detection measurements really need to have high sensitivity. And this is because the biomarkers that we're usually interested in to detect the disease are found in very low concentrations and really usually very complex matrices like blood or tissue. And so it's for this reason that we have to go to surface enhanced Raman spectroscopy to do these measurements. So surface enhanced Raman, or SERS, is a variation of Raman spectroscopy, but instead of our normal kind of sampling setup that we have on the lower left side of the screen, instead we adsorb our sample to some sort of metal nano substrate, which is usually fabricated from either gold or silver. So the enhancement that we see here results from the amplification of light by the excitation of what's known as a localized surface plasmon resonance, or LSPR. And so our LSPR is just kind of an oscillation of the valence electrons that make up our metal. So you can kind of see that drawing of that on the right there. And so this enhancement that we get can be, again, on the order from 10 to the third to 10 to the seventh, even greater, depending on the SERS active material that's used. But this enhancement really allows us to reach trace level detection of biomaterials. And so this is research that's presented in collaboration with Mark Porter's group from the University of Utah. So to detect tuberculosis biomarkers, they came up with what's called a SERS sandwich-styled immunoassay test. And this will detect and potentially quantify the amount of the tuberculosis marker in human serum. And this type of test is based on the binding of the antigen to the antibodies. So to prepare this test, they first had to load the capture antibody. So it's the anti-antigen MAB, the red antibody on the upper left part of the screen. So they had to load that onto the glass and gold film support using a coupling agent, which is the DSP. And then the second part is they had to make what's called an extrinsic Raman label, or an ERL. And so for this, they coupled gold nanoparticles, and that's what's going to give us our SERS enhancement. So they coupled the gold nanoparticles to the Raman reporter molecule, which in this case is DSNB. That's the little green picture that's shown on the lower left-hand corner. And then added the secondary antibody to that as well. So the nanoparticle, the Raman reporter, and the secondary antibody all make up that extrinsic Raman label. And so once the sample is loaded, that antigen can be captured or sandwiched between the capture antibody and the secondary antibody if it's present. And then the SERS measurement is made, and we're able to relate either the presence of the Raman reporter molecule and the amount of it that's present to either the presence and also the amount that's present of the antigen. So the idea is that if there's no antigen present, then the extrinsic Raman label won't bind won't be able to bind, and so we won't observe any of the signal from the reporter label. So this allows for really selective detection measurements of these type of antigens. And they used a handheld Raman system specifically to do these measurements because this allows them kind of the portability that they need to apply this measurement at points of need outside of the laboratory. So the detection levels for the antigen are very low at nanogram per milliliter level. And we can also potentially quantify the amount of the antigen that's present in a new sample from a patient using this type of analysis. So we can create a calibration curve like the one shown below here. Okay, so those are just a few examples of some of the applications that our systems have been used for recently in the biomedical field. So now I will turn it over to Adam, who will give us kind of an overview of the Metrohm Raman Spectroscopy portfolio.
Thanks, Kristen. I appreciate it, and thank you for that overview of all the different, of some of the different types of applications. And I know there are more out there. The literature is filled with people deploying Raman spectroscopy in innovative approaches. Ultimately, when we talk about spectroscopy, the reason that we talk about spectroscopy, and part of the reason that it gets so much, is getting so much attention, is because of the benefits that spectroscopy can bring, less in the research and development phase than in the ultimate phase of when you actually have an analysis, a diagnostic built out. And that's because spectroscopy delivers speed, and the technique itself is versatile, and with a little bit of customization, such as with the SIRS work, you can see you can really develop application-specific approaches with spectroscopy. And then, of course, spectroscopy can be made compliant, which, if you are going to be in the medical space, you certainly have compliance issues that you must overcome when you're putting out a tool for the point of care. And if you were listening, one of the things that Kristen said was, in discussing the cancer margin analysis, and that's where typically, right, she said it takes about it would take a couple of weeks for the pathologist to move through the sampling of the tumor specimens and say, hey, did you get it all or not? And with spectroscopy, you could potentially do that much, much faster, either via some automated imaging or some very quick testing there at the point of care, just to make certain that you've gotten everything out. And that is the idea of extending the value of other laboratory techniques. So you can take the laboratory technique, in this case, say the trained pathologist, and to a large degree automate that into a spectroscopic chemometric method. And that's going to increase the precision of the results, because it eliminates the judgment call and decrease the error potentially associated with that analysis or other tests. At Metrohm, we offer a range of spectrometers. So we have handheld spectrometers, which were highlighted with a SIRS application. And these spectrometers are typically used around the idea of raw material verification in the biomedical space, but we see an increasing desire to deploy these into research because they are very, very simple to use and can be scaled up into dedicated analyzers for biomedical applications right away. We also have laboratory spectrometers, which are the perfect flexible tools for doing these investigative studies. And again, you could eventually scale them up into a device. And then we offer a line of process spectrometers as well, so that if, let's say, we're talking about that vaccine production, we can monitor the growth of the bacteria or the viruses, whatever it is. I guess it's vaccine, it's viruses. We can monitor the production process of those materials. Just taking a quick look at the benchtop, the laboratory Raman spectrometer family that we offer through our B&W Tek brand, which is what we've highlighted here. There are 4 different families of systems, if you will, covering different excitation wavelengths, different laser powers, as well as ranges and resolutions. And these systems are all lightweight, all durable, and all portable, so that you can take them easily from the laboratory where you're doing your R&D work and then move them into those first rounds of studies, say, in hospital. And as Kristen pointed out, the advantages of this type of system are the fiber optic coupling, because you get the flexible interface options. So whether you want to talk about just monitoring something in, say, a cuvette, whether we need just to increase some stability as we're measuring over a sample, because we don't want to hold it by hand, although certainly you can hold by hand for a lot of things. Microscopes, probes, flow cells, so if we are monitoring what's happening in, say, that production environment, you can monitor the Raman signal in a live phase. And then customized probes as well, so that we can go in and talk about measuring, perhaps not with a microscope, but measuring at that micro or near micro scale with specially designed optical probes. So you can get data from whatever the biological sample is that you're interested in. And then from a software perspective, key is we offer several different kinds of software that can be used. So whether you're talking about just early stage development, where you just need to explore what's happening in the system and get Raman data so that you can start manipulating it and crunching it and seeing, is there interest there? So software for development, software for analysis, either through our Vision software platform or through integration with Unscrambler. And then operationally, you can take what you develop in the analytical software can then be taken and deployed in routine operation for analysis that can be done truly at point of care with minimal training, or analysis of, again, a process that's underway. So to summarize, we think we've shown where a number of Raman systems can be used to produce information that is relevant for development of diagnostic tools and even, and again, monitoring tools. And the big strength of Raman spectroscopy in this space is that the spectra are information rich. You saw that with the very narrow bands in Kristen's slides. And it is free of water interference, unlike FTIR and near-infrared spectroscopy. You're not going to get the interfering signals from Raman, which lets you then go in and look at the molecules of interest, particularly. And it's relatively small amounts of sample prep for this. And then, of course, we can combine those, and that's what you're seeing in some of the data. And a lot of the research that's going on is this combination of spectral data with clinical data, with chemical understanding of what's happening to produce a robust classifier under a quantification method. So with that, I thank you for your time, and we are happy to take your questions.
Thank you, Kristen and Adam, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. And as a reminder, any questions that we are unable to answer today, they will be addressed by the speakers via the email address that you provided at the time of registration. So let's get started with our first question. You mentioned Raman Spec has no interference from water. What about interference from biofluids like blood or urine?
Sure. So that's a good question. So we don't have interference from water, but other types of solvents or biofluids, like the ones mentioned, will have some Raman signal from them. So that's why it's sort of dependent experimentally. If it's just the blood or the urine that we're trying to detect or compare to something else, then we can use that signal. But if we're trying to measure the very small amount of something that's either in the blood or in the urine, that's usually where your surface enhanced Raman measurements, where you have a little bit more sample preparation, will come into play.
Perfect. Thank you, Kristen, for that clarification. So moving on to question number 2. Would this work for metabolomics? For example, analyzing media samples with metabolites produced by cells or bacteria?
Yes. So monitoring of cell growth with Raman is a very sort of popular biopharmaceutical process monitoring for Raman spectroscopy. So measuring basically the different metabolites that are produced by the cells as they're consuming the media. And so this is usually done building some sort of chemometric model for quantitative analysis, where you have some known concentrations to build your standards off of, and then with the Raman measurement, you can do it just directly in the vessel, so you don't have to sample from the vessel itself, and then also that's a non-destructive measurement, so you're not altering the actual sample itself.
Wonderful. Now, our next question. Is there a limit on the excitation wavelength to use in Raman spectroscopy?
So for excitation wavelengths, we normally have 532 nanometer laser, which is our lowest wavelength laser, and our highest one is our 1064 nanometer laser. So we have 532, 785, and 1064 nanometer lasers. There are other types of lasers that we can use to do Raman spectroscopy, like gas lasers and UV lasers as well. They go down into the UV, but those tend to be a little bit more sort of intensive in terms of experimentally. So for the semiconductor lasers, we have our 532, our 785, and our 1064, which should cover most very common applications.
Wonderful. Now, is there any integration of the lab Raman spectrometers with Metrohm's potentiostats?
Yeah, if I can grab that one. Yes, these laboratory spectrometers can be integrated with our potentiostats. And if that's not quite the way you want to go, we also offer a line of spectrometers that are integrated with with some of our other electrochemistry products. So we have a full line of spectroelectrochemistry.
Perfect. Thank you so much for that, Adam. Now, our next question, what about fluorescent signals in some other applications? Kristen, go ahead, Adam, please.
Yeah, I was going to say, if I think the question was, what about fluorescent signals in some other applications?
Yes.
I guess their question would be, what is the application? Is the fluorescence a signal or is it an interferent? So when we're talking about Raman spectroscopy, we're often thinking of fluorescence as an interfering signal as opposed to an actual signal that we are interested in getting. But certainly you can use fluorescence as an orthogonal detection mechanism.
Wonderful. Okay. We want to thank the audience. We've gotten so many good questions in, so let's look for another one. Okay, here's another one. How about systems which are largely complex, like environmental samples, for example, soil? Is it possible to decouple composition and interactions?
Sure. So, yeah, for very complex samples like soil, again, it's going to kind of depend on what you're looking to measure here. A lot of times in samples like soil analysis, we're looking for more elemental information than molecular information quite often. And so Raman may or may not be the best tool for that. I would say that probably an elemental technique like LIBS, laser-induced breakdown spectroscopy, is better for that. But certainly there are, in very complex environmental samples, there are sample preparation tools that we can use to sort of sort out the actual analyte that we're looking for. So, for instance, microplastics come from a very complex environmental sample, but there's sampling methods in that type of field that can be used to extract the plastic material.
Yeah, and if I can weigh in on that one, just because I happen to, if I go back to my undergraduate days, I can remember doing a project on this. And we actually were able to successfully study the way some humic acids in some soil samples out of somewhere in the western United States, how they were actually changing as they were complexing and exposed to different saline solutions via their Raman shifts. So we could actually watch some of the peaks, the fine peaks, shift around a little bit and infer what kinds of some of the interactions that were occurring. So you can get in there and see that level of detail because the spectral resolution spectral information is so high.
Perfect. Thank you so much, both Kristen and Adam, for that answer. So it looks like we've got time for about one more question. Now, can you use a laser-directed beam to reflect off of skin and measure the Raman effect?
Yes. So this measurement is definitely possible to detect, especially certain types of tissue. It can also be used to measure if there's potentially a skin cancer tumor. The experimental challenge here is making sure that we reach kind of a deep enough penetration of the laser through potentially that kind of upper thick layer of the skin. So we actually do have a system where it uses a very defocused laser spot size that's able to penetrate deeper into the skin to not just get signal from that very top part, but really probe into the kind of localized environment beneath that. So that's our ST probe with a very large spot size, about 4 millimeters, that's able to accomplish that.
Wonderful. Thank you so much. Before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information that you provided at the time of registration. We would like to once again thank Kristen and Adam for their time today and their important research. We would also like to thank LabRoots and our sponsor, Metrohm USA, for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay. That's all for now. Thank you for joining us, and until next time, bye-bye everyone.